everyone, welcome back to the DNA Lab Short Read Workshop. In this video we're going to go over calling chip peaks using the tool max. As you learned in the previous video, the purpose of ChIP-seq is to identify the regions of the genome which are occupied by our protein of interest, whether that's a specific histone mark or a transcription factor. Previously, you've learned how ChIP-seq libraries are prepped and how those libraries are then QC'd and mapped to the genome. In this video, we're going to go over the next step of the ChIP-seq analysis pipeline. This step involves identifying each enriched region of the genome in a process known as peak calling. Let's talk first about the challenge involved in peak calling. Take a look at these two IGV tracks. On the top we have our chipped library track, which should show enrichment of signal at regions where our protein is bound to the DNA, and below that we have our input library, which ideally should show no enrichment of signal across the whole genome. In reality though, data is messy, so our input library isn't entirely flat, and our chip signal also shows a lot of background noise throughout the whole genome. So while some peaks might be easily identifiable by eye, some are a little bit harder to distinguish. For example, let's take a look at these regions in the red boxes. Each of these shows more reads in the chip library compared to the background, but is it significantly enriched enough to be considered a peak? The two in the middle look like they're good candidates, but there's also some signal enrichment in the input file at those regions. And then another potential peak on the far left is only slightly enriched above background. Is that considered enriched enough to be called a peak? Furthermore, how should we determine where a peak begins and where a peak ends when these regions are in a sea of this background signal? To answer these questions, we need a statistical approach which can rigorously test all these regions throughout the genome. This test should ideally separate real peaks from the background noise that might just resemble a peak by random chance or because of some unforeseen source of bias. Now, there are many peak callers which can perform these statistical tests, but the most common one is known as MAX, or Model-Based Analysis of ChIP-seq. MAX will compare the enrichment of signal in a given region to the local background and to a specified control file. The relative confidence of the peak call is represented by a Q value that MAX assigns to each region, and the output of MAX will be in embed format. Now, we'll go over the specifics of this file format in more detail after we've run the program, but the important takeaway is that MAX provides us with a file that contains the exact genomic location of every enriched region, along with a confidence for the peak call. Now, the math underpinning MAX statistical tests and how that model is actually formed are beyond the scope of this workshop, but you can visit the GitHub at the link below if you're interested in learning more about the program. There's also a link on that website to the original publication, which I'd encourage everyone to read to understand MAX in more detail. Now, a good MAX run requires four things from the user. First, you'll need your mapped chip library as a BAM file. Second, you'll need your input library that's also going to be in BAM format. Now, you've learned how to generate these files from raw FASTQ files in week 1, so if you're still feeling shaky about how to do that process, make sure to revisit those sections of the workshop for a refresher. As a side note, MAX can also be run using a treatment and control file if you're interested in doing differential peak calling between conditions, for example. The tool runs in the exact same way regardless, so for today I'm just going to talk about running MAX with a chipped library and an input library. Now, the next things we need to specify is whether MAX should look for broad peaks, like a histone mark, or narrow peaks, like a transcription factor. And then lastly, MAX requires us to indicate how large in base pairs the genome is. This step is really crucial to correctly calculate the statistics for the peaks, so remember, if you're using a different genome or if you've truncated your files, you'll need to adjust this portion. For example, if I were only calling peaks on one chromosome, like chromosome 21, I should only specify the size of chromosome 21 as that genome size, rather than specifying the whole genome. There are, of course, many other control parameters which you can use to fine-tune your output. We're not going to go over all of those parameters in this video, but again, you can visit that link below if you want to learn a little bit more information about all of those parameters and how to fine-tune the process for your specific data. Now, MAX is quite a lightweight tool, but we're still going to be using it on the AWS because that's where our BAM files are and because we can make use of the extra memory there. So let's go ahead and take a look at a typical MAX script now. So here I am on my terminal. I've already logged into the AWS, and I'm currently in my scripts directory here. If I print the directory, you can see that. And I store my BAM files of interest in this directory right here. You can see I have a chip BAM file and an input BAM file. Now, I've already written the scripts, and we can go over some of the parameters here. So let's go ahead and enter that script using Vim. Now the first step, as usual, is to set all of our Slurm parameters. So the top line of the script will specify that this is a bash script using this language here. And then I'm going to set my job name, max2. I will then set my mailing parameters, as I've done so here. Then, as I mentioned, Max is quite lightweight, so we only need one CPU in this case. I'll set a wall clock of only 20 minutes. I'll use the short partition, and I'll use a relatively small amount of memory at 4 gigabytes. And of course, I'll also specify my output and my error directories at the paths here to make sure that I can use those files for troubleshooting later. Next, we'll module load Max. Remember, if you're unsure about which versions are available for your tool, you can always search that using module spider. So if I didn't know which path to use for this Max module, I would use module spider Max on the terminal, and that would tell me that this is the module that I should load for Max. I've already loaded it in here using this version, 2.1.1. And so now, once I've loaded that module in, the program will be able to run in this script here. Now to actually run Max, we're going to use the call peak function here. So I'll write max2 call peak. Then I'll use the -t flag to specify my chip file. So I'll give it the full path to that chip BAM file. Then the -c flag, I'll give it the full path to that input file. Then I'm also going to specify an output directory using this outdir flag. And I'll specify it for this, again, using the full path. And then we come to that all-important genome size flag. So I'll use -g, and then I'll specify the genome size in base pairs, and I can also use scientific notation here, which in this case for my file is a value of 2.7 times 10 to the 9. So if I wanted to change this to broad peaks, I would also add the broad flag here. Now in this case, this file is for a transcription factor, so I'm not going to include that broad flag. And Max just has it generate narrow peaks by default, so you only include that broad flag if you are going specifically for broad peaks, but if you're going for narrow peaks, then you don't need to include any flag here at all. So once I've double checked my input and made sure everything looks good, I'll go ahead and save this script and exit Vim. And then I would submit this script the same as I would submit any other sbatch script using that sbatch command. And I would hit run and let that run in the background. Now, for the purposes of this video, I've already run that script, so now we can go ahead and take a look at the output. So I'll change directory to my peaks file here. And then I can list the contents of this directory using ls. Now you can see that we have a few files here. The model.r file here on the left is info about how Max actually built the model, so it's not necessary to view that file in detail unless you're trying to troubleshoot. This peaks.xls file contains all of the peak information in an Excel format, so if you prefer using that program for viewing your data, you can load that into Excel. And then you've got these two other files, the summits.bed file and the narrow peaks file. This summits.bed file is another extra file that only contains the summit of each of the peaks. And then you have this the most important output file, which is this .narrowpeak file here. Now, as I mentioned before, this .narrowpeak file is going to be in bed format, which is a standardized format for a lot of these genomic files. So let's go ahead and take a look at that now. We'll print the first few lines using the command head, and then we'll just use that on that narrowpeak file. So the first 6 columns, as I mentioned, are in what's called bed6 format. This first column is the chromosome location. The second column is the start position of the peak in base pairs. And then the third column is the stop position of the peak. The fourth column is a name for the peak that the tool generated for it. The fifth column is the score for the peak. And the sixth column is the strand. Now, because we're not working with stranded data, this strand value just appears as a dot. Now, the 7th through 10th columns are actually Max-specific columns, so they're not specific to the bed formats, but they are specific to the Max output itself. The 7th column is the relative enrichment. You can think of this kind of like a fold change of the signal above background noise. The 8th and 9th column are the negative log 10 of the p-value and q-values respectively, so these give us an idea of the confidence of the peak call. And you may notice that the score from the 5th column is actually just the same as the negative log 10 of the q value multiplied by 10 and then with the decimal truncated off. So here 5.16, multiply that by 10 and truncate the decimal, you get 51. 9.8, over here you get 98. So these are relatively the same value, and again, higher scores just indicate that we're more confident in that peak call. Now Max has already filtered out any low confidence peak calls that don't meet our minimum value. By default, Max uses a Q value of 0.05, although you can adjust this freely as needed. And all of those low confidence peaks have already been removed from this file. But if we wanted to further truncate this file to higher confidence calls, we could of course filter further on these peak calls here. And then finally, in this last column here, we have the location of the summit from the start point. So the summit is 119 bases away from the start position at 10,061 for this first line, 118 bases for the second line, and so on and so forth. So that's the basics of these narrow peak files, and as I mentioned, these are bed files, which means that we can actually load them directly into IGV to view them visually rather than just scrolling through the text here. So I've transferred this peak file to my local computer, and now I can switch over to IGV. And I can load that in along with my chip TDF file and my input file TDF. Now it's always a good idea to scroll through a few of these peak calls along with your TDF to make sure that the peak calls look good and make sense visually. So, and if you know any of these genomic regions that should be bound by your transcription factor, like if you know a target gene or an enhancer, it's a good idea to specifically go to those regions and look at the chip file and the peak calls as a sanity check. And that's what I'm looking at here. You can see in this window that there are 3 peak calls. The peak call on the left aligns very well with this enriched signal here in the chip file. And same with the one on the right. There's clearly some signal enrichment here, although it's less strong than the one here on the left. There's also another peak here in the middle, which is a little bit less convincing, although it's still well above background. And if we wanted to continue this process, we could just scroll through IGV and check out other peak calls and make sure that they make sense with our data. Now, if you're seeing a lot of potential false positives or false negatives, it's worth going back and rerunning Max, adjusting some of those parameters and fine-tuning it to more closely fit the particulars of your data. And again, remember to visit that Max webpage to learn more about that fine-tuning process and what other parameters you can use. Now, these peak calls are going to be the basis of our analysis going forward, so it's really crucial that they're of high quality. And of course, in class, we're going to go over a little bit more specifically how to perform some QC checks on these calls, and then also how we're going to be able to use them for things like motif discovery or tying them to target genes. And those are the basics for today for running Max on your chip data. I encourage you again to visit their webpage regularly to keep up with the data and also figure out what might have changed with some new releases and if you need to adjust anything to run it on your data specifically. Now in the next video, we're going to go over using our peak calls for motif discovery using the meme suite, and so I'll see you then.